Hey everyone, Armin here from Hex. Today I want to walk you through a drug response study where I captured 300 participants with type 2 diabetes and monitored their HbA1c levels over a 6-month treatment plan. To start out on my analysis, I captured their age distribution and the split of the cohorts between their male and female counterparts. Specifically on the HbA1c levels, these are blood glucose levels that the lower the level, the better treatment we have of type 2 diabetes, generally speaking. So in this case, I looked at post 6-month treatment plans after this medication has been applied. And we noticed that about half our individuals in this cohort actually improved state. About 100% had no change, and 27 had actually worsened effects, with a nice little write-up here for the reader. I split this out by the gender as well, and we noticed we actually have a little bit of an uptick here in improvement on the female cohort. Nothing to indicate too much significance, but something that we want to take further if we're doing this cohort or analysis with a new set of individuals. Next, I paired this information with some genomic associations. Specifically, I wanted to target which chromosomes actually have some impact and on type 2 diabetes, and how do they respond based on this new drug. So here I can tell that chromosome 11, 4, 19, and 7 actually have indications of very strong drops in HbA1c levels just in a matter of 6 months. This is something that, one, can give me competitive advantage against my competitors, and two, can prompt better patient care, where I can actually have a better targeted approach and better recommendations for people that are predisposed with these chromosomes. So I can target specific alleles and get even more granular with this. Next, I looked at their clinical significance by pulling in data from the ClinVar database. It could be any database that you want to bring in, all powered under the hood by Snowflake, and I'll show that in a moment. But here I can actually look at specific risk factors or maybe benign indications of people that have type 2 diabetes or some genomic dispositions to type 2 diabetes. And in this group, I can notice that of folks with high risk factors, we actually see 4 of them have strongly improved performances, with 3 having no change. So we're at least not getting any worsened changes on folks that have very bad type 2 diabetes levels. Lastly, I do the similar kind of spread here amongst the genders. And to show you how this whole entire project was built, I'll jump over to what is effectively under the hood behind every single Hex project, and that is a notebook. Where folks coming from, say, Jupyter, they will be very comfortable in this environment, where a technical persona can come in, work with SQL, with Python, with Snowpark, add text cells, add native Hex visuals with our charting capabilities, all in an effort to create this final data app. And then finally, you can publish and share those changes out. If you'd like to explore this app yourself, or potentially go try it out in your own Hex environment, I'll leave a link to the top right. Thanks for watching.